Welcome to the 2022 University of Georgia 3-Minute Thesis Final. My name is Jack Hu, Senior Vice President for Academic Affairs and Provost at the University of Georgia. Our graduate students are at the forefront of innovative research and scholarship. It gives me great pleasure to share their outstanding work with you. Today's competition will feature the work of students from disciplines across campus. It's an excellent representation of the quality and breadth of scholarship at UGA. Congratulations to all the graduate students for being selected for the 3-Minute Thesis Final. Now I would like to introduce our Master of Ceremonies, James Hathaway, who will tell you more about the competition.
Well, thank you very much, Provost Hu, and let me also welcome everybody to the 2022 University of Georgia 3-Minute Thesis Competition. This is the 11th 3-Minute Thesis Competition here at the University of Georgia, a fact that I am particularly proud of. I've had the pleasure of working with students and faculty and staff as part of this competition for a number of years now, and I'm always delighted to see so many of our very best and brightest competing here on the stage. Speaking of the stage, you'll notice that this format is a little bit different from years past. Sadly, we are still not 100% in person. But we are pre-recording things so that everybody can watch the competition all at once. And you can all have a sense of being here, even if you can't actually be here in body. So I'd like to begin by going over a few bits and pieces of the competition, particularly the rules. Competitors are allowed only one static PowerPoint slide. It can't have any transitions or animations or any kind of movement. They're also not allowed to have any additional electronic media. There can be no music, no video files. They can't have any additional props, so they can't use costumes or musical instruments or laboratory equipment. They're limited to 3 minutes maximum, and any competitor who goes over that 3 minutes is disqualified. Presentations are to be spoken only, so there's no rapping, there's no beat poetry, there's no poems. Presentations have to be given from the stage, and they are considered to have commenced when the presenter starts their presentation, either through movement or through speech. So the final thing is the decision of our judging panel is final. Speaking of judging, I want to give you a sense of how competitors will be evaluated for the competition. The first broad category is comprehension and content. The presentation has to be provided in a clear, has to provide clear background and significance to the research question. The presentation has to clearly describe the research strategy or design and the results or findings of the research. And the presentation has to clearly describe the conclusions, outcomes, and the impact of the research. But equally important is the category of engagement and communication. Presentation has to be delivered clearly and in a language that is appropriate for an audience of non-specialists. The slide has to be well defined and it has to enhance the presentation, and the presenter has to convey enthusiasm. They actually have to seem like they care about this a little bit. And the research has to capture and maintain the audience's attention. Now, our competitors this evening will be evaluated by 3 judges. The first is Ms. Tracy Cook, the Executive Vice Chancellor for Strategy and Fiscal Affairs at the University System of Georgia. The second is Mr. Nicky Jones, Assistant Manager, Unified Government of Athens-Clarke County. And finally, Mr. Neil Quirk, the Chair of the University of Georgia Foundation Board of Trustees and partner at Quirk & Quirk LLC. Now, I want to remind everybody that you will also have a chance to vote on our competitors. We're watching this on Facebook, so if you look down here, I guess, there's a section in the comments or a link in the comments where you can go and vote on your favorite competitor, and the person who receives the most votes will receive the People's Choice Awards. And so with that, I think we're ready to begin. Good luck to all the competitors. All right, our first competitor this evening is Lauren Scrow, and her talk is titled Missing Dust Around Red Dwarfs.
Space, the final frontier. One of the reasons it remains as the final frontier is, well, one, because of Star Trek, but two, because it's so incredibly immense. So huge, in fact, that the most recent telescope survey of our entire sky, called Gaia, looked at 1.7 billion stars. And yet, that's only 1% of all stars in the Milky Way. Now that means our home galaxy contains over 200 billion stars, and about 75% of these are red stars that are cooler and smaller than the sun. Astronomers call these red dwarfs. And even though these stars are the most common in the entire galaxy, there's something strange happening around them. They're missing their dust. Yes, dust, space dust, if you will. You see, stars are born with leftover gas and dust that didn't get made into the star itself, and that leftover primordial material orbits the star in a disk. If we're lucky, materials in that disk can stick together and begin to form baby planets. Once planets are fully formed, they can collide with themselves and other objects and create dusty debris of their own. This final stage of the disk is called the debris disk, and we actually have two debris disks in our own solar system, the asteroid belt and the Kuiper belt. Now we see this first stage of disk around red dwarfs. We see that leftover primordial material, and we see the second stage, the forming planets. We even see the mature planets themselves, but we don't see the debris disks around them. Now why is that? Well, astronomers have a couple of ideas. The first is that maybe red dwarfs form planets differently than their hotter cousins, and that's why we don't see these disks. They simply don't exist. Others think that these debris disks are there, but we just can't see them with our current technology because they're hard to observe. But that's where I come in, to help settle this debate and hopefully bring a little bit of light to planet formation around the most common stars in our galaxy. I've used that new Gaia database to look at nearby red dwarfs, the most anybody's looked at one time before, so I should be able to tell whether this lack of red dwarf debris disks is real or imagined. So, how do I find that dusty debris around red dwarfs? Well, I look at these stars in the infrared. You see, we have telescopes geared towards detecting this wavelength of light, and that helps us find dust. Because as dust is heated up by its host star, it gives off heat of its own, which is then detected as infrared light. So if I'm looking at a red dwarf in the infrared, and it's brighter than I would expect, just from the star itself, I know that there has to be dust. So, what did I find in this grand search for red dwarf debris disks? Well, out of tens of thousands of nearby red dwarfs, I found 33. It's a little anticlimactic, right? But it's actually incredibly important. You see, with a huge sample size, we still see a very small number of red dwarf debris disks, furthering the argument that they really just might not be there. And we also found new debris disks. Because these are so rare around red dwarfs, this is actually incredibly exciting. So, these will still need to be confirmed with follow-up images, and hopefully we can use a little telescope you may have heard of called JWST to do exactly that.
Our next competitor is Leah Dutton, and Leah's talk is called Prevention, Early Recognition, and Intervention Techniques for Injuries in String Players.
A few years ago, I went to see famous violinist Sarah Chang perform in concert here at UGA. Despite her incredible performance and her gravity-defying backbends while wearing 4-inch heels, the thing that I remember most about that concert was that every time she stopped playing, she would grimace and rub her left hand in pain. String musicians have some of the highest injury rates of any profession. Although orchestra players may appear polished and professional while on stage, 40% of them are currently in pain, with a lifetime injury prevalence of around 90%. These injuries aren't simple, trifling causes that can be easily resolved with a few weeks of rest. We're talking decades-long battles with things like carpal tunnel, tendonitis, and thoracic outlet syndrome. In fact, 60% of professional musicians will become so injured that it's physically impossible to play their instrument for some period of time. Remarkably, the majority of these injuries are avoidable. Studies have found that 94% of the injuries sustained by musicians are considered preventable. So why are these injuries still having such a devastating impact on the community? Unfortunately, string musicians are taught from a young age to sacrifice their well-being for the greater good of a performance. Pain and injuries are never talked about inside the classroom or the concert hall, and students graduate from university thinking that pain is a normal part of music performance and spend years trying to conceal and suppress their worsening injuries. And just like that, simple and easily resolvable conditions turn into complex, chronic conditions that are incredibly difficult to treat, forcing 10% of musicians to abandon their careers and never perform again. My research seeks to break this cycle of isolation, negligence, and injury within the music community. I've designed a college course and textbook for undergraduate string students that explores the causes and consequences of injury in conjunction with prevention and treatment strategies. It is an accurate and informed WebMD for string musicians, that string musicians can look up their symptoms or causes of injury based on instrument, body part, or even condition to learn more about their symptoms, causes, and treatment strategies. I collaborated with physical therapists and other medical professionals to create symptom and testing flowcharts, sample rehabilitation timelines and strategies, and exercises to increase stamina and improve performance outcomes. This resource also provides conservative treatment measures for musicians who aren't able to see a doctor right away, but encourages early medical intervention through health education. For example, students who learn that the turnover rate of tendon tissue isn't just 1 to 2 months, but is actually closer to 1 to 2 years, will be far more likely to take their injury seriously and react at the first sign of trouble. And early intervention is the most effective treatment for all of these conditions, making easy access to this information an essential component of string pedagogy and future achievement in music performance. Thank you.
Our next competitor is Colleen Sedney. Colleen's talk is called Virtual Memory, How Babies Pass the Test.
Has anyone heard about pertussis lately? I mean, big whoop. And that's right, it is a big whoop. Whooping cough, that is. Bordetella pertussis is the bug responsible for whooping cough, which kills tens of thousands of babies every year. This bacteria causes disease in the lungs, which results in a distinctive cough symptom. Now, we might ask ourselves, why babies? Don't adults get sick with and die from whooping cough too? And the answer is not really. Whooping cough is mainly a disease that infects babies, and most people think it's because they have an immune system that just doesn't know what to look for. It's like taking a test that you haven't studied for. The immune system doesn't know what to look for because it hasn't seen any bugs yet, or taken the test. But if that were the case, then babies would get sick with and die from almost every bug they ever saw, which they don't. Now, I'm sure that everybody has that one friend that never studies but always gets a good grade, right? Well, we have observed something similar in babies to help them fight bugs. Babies have a pathogen-fighting immune cell called virtual memory cells. Now, even though this cell has never seen a bug before, it is able to act like it recognizes it and immediately charge into battle. We think that it's these virtual memory cells that protect babies from most bugs, like we can see on the left. But why did it take us so long to figure this out? That's because a lot of our understanding of the human immune system comes from using mice as a model. In human babies, it can take weeks or months for their actual memory cells to learn how to fight a bug. But in mice, it can take only days. So it's been difficult to figure out just how these virtual memory cells are able to see and fight a bug that they have never studied before. My research has focused on developing a new model system to pinpoint the best age that we can figure out just how these virtual memory cells are able to see and fight a bug. Oh, God. But now that we know about virtual memory cells, why do babies still get and die from whooping cough? My research suggests that the whooping cough bug makes a toxin that essentially acts as a trick question that confuses the virtual memory cells in babies, making them fail the test. But the actual memory cells in adults have studied for this, so they are able to recognize the toxin and fight the bug. Our goal is to figure out the perfect cheat sheet that we can give to these virtual memory cells to get around this trick question toxin. If we can do that, then we could save thousands of lives and prevent anyone from failing the test. Thank you.
Our next competitor is Shrina Pradhan, and Shrina's talk is titled Salt Glands. When in doubt, sweat it out.
So the Earth's surface is 71% water, and of it, 97% is salt water. And salt water means that it is undrinkable. Not only that, we can't irrigate our croplands with it either. Cereals, vegetables, fruits, you name it, they are all susceptible to high amount of salt and are going to start dehydrating and dying upon exposure. It's like you're thirsty and then you're grabbing a glass of seawater to drink. It's going to leave you with such a bad taste in your mouth and dehydration. And if you think about it, since most of Earth's surface is salt water, a lot of Earth's land is bound to get salinized. And that is what a salinized land looks like. That land is uncultivable. And this happens to a point where every year we lose cultivable land that is about the size of state of Georgia to soil salinization. What an alarming loss, considering the population is only growing and we need more food now than ever. So, in order to make sure that our food production does not succumb to this problem, I look at the genetic components of salt tolerance in plants. Now, what if I tell you that there are some magic plants that we call halophytes, that literally translates to salt-loving plants, that can withstand this high amount of salinity that is almost as high as the ocean water itself? And not just that, they're very closely related to some of the cereal crops that we consume as staples. Let's call them first cousins. You know, the same family. So a mechanism of salt tolerance in these plants, out of very many of course, is secreting that excess amount of salt that they have through a structure known as salt glands that is on top of the leaf. And you can see that right here, the salt gland. It is no bigger than half the diameter of a human hair. So what happens is the salt from the water tries to travel up a plant's vascular system, which is kind of similar to our blood vessels, meaning that they go into every single cell, right? So every single plant cell has salt. And what these salt glands do in halophytes is they collect all of the excess salt and then try to secrete it out into the leaf surface. Or, excuse me, they try to sweat it out into the leaf surface. And you can see that right here in the picture. And here's the good news. We know some of our cereal crops also have these salt glands, only they don't work. But I'd like to say they don't work yet, because I aim to study the genetics behind the salt glands in these halophytes that do work, so that we understand it to a point where we can make the ones in the cereal crops work too. And that's going to help us grow our cereal crops in land like these that we thought we lost to salinization. That's also going to help us use brackish water, which means like kind of salty water, to irrigate our food. That's going to save us so much resources. Now, I want to leave you with this. Salt glands are just one part of the solution, and there is so many other ways that we can breed a plant to become more tolerant. So please take all of this with a grain of salt. Pun intended. Thank you.
And our next competitor is Eleanor Hare. Eleanor's talk is titled North Avenue Redesign.
If you walked a certain street every day, multiple times a day, and you heard that the government was completely redesigning it, wouldn't you want to be involved? Reimagine North Avenue is an Athens-Clarke County project which aims to redesign North Avenue from the Tin Loop to the Greenway to make it more accessible for pedestrians. The county has yet to receive funding from a federal grant, so the project is still in preliminary stages. And this is a fantastic opportunity for community-driven, locally planned design. I am working with the communities around North Avenue, surrounding the project site, to conduct this redesign. I hope to engage stakeholders and local residents in the design process to determine what values they hold, what needs they have, and how this redesign can best serve them. Many residents around North Avenue are living below the United States poverty line. 15% of these residents do not own a car, meaning they rely on biking, busing, and walking to get around. There's also a large homeless community in this area because of the many homeless shelters that are along the street. Arch, Advantage, and Bigger Visions are all walking distance from each other along North Avenue. It is imperative that everyone who needs it has safe access to these buildings. Many of the guests of these shelters are sick or have injuries, and they need easier pathways to reach these resources. I will be working with these shelters, along with schools and places of worship, to conduct design activities. And these design activities are aimed to gather data on people's wants and needs for the North Avenue redesign. Now, to get the community involved in the design process, people have to be interested in participating. Surveys and community meetings rarely get the proper attendance or attention needed to be beneficial. So I had to get creative. Games. Games are an easy and fun way to communicate ideas, and everyone can participate despite different reading levels, mobility issues, and design skill. I will administer a variety of design games, including Build Your Own Model, Landscape Tinder, and Storytime. And this will inventory the community's character, their values, and their ideas. I will then take these ideas and merge them into one final design solution. And this design solution, the North Avenue redesign, this is the final product. But the craft of getting there alongside the community is the thesis. I hope to uncover the challenges and the benefits of working alongside a community in this manner. And it's my hope that this process will be used as a model for future public designs. Thank you.
The next competitor is Smita Rawal, and Smita's talk is called Naloxone, Bringing Back from the Dead.
65,700. That's how many people died in the US in 2021 from the opioid overdose. Today, 180 people will die. Opioid overdose does not discriminate by age, gender, race, ethnicity, or your political view. It all ends the same way. You stop breathing. Your brain cells run out of oxygen. Within minutes, your heart stops beating, and then you die. Who is at risk? Normal people like me and you. Your neighbor who is prescribed pain medication after a knee surgery. Or my cousin on pain medications who decides to drink a bit more alcohol to celebrate Bulldog win over Bama. Now, what if I were to tell you that there is a complete antidote that is effective in reversing the effects of opioid overdoses? Yes, we have a life-saving medication called naloxone, commonly known as Narcan, available from pharmacies. If a person is not breathing and unresponsive due to a possible opioid overdose, step number 1 is to call 911, and step number 2 is to administer Narcan. Narcan can be sprayed through the nostril, and just like turning on a light switch, a person can be awake, walking, and talking within mere seconds. If this antidote is so effective and fast-acting, then why isn't it available to everyone everywhere? 2 reasons. First, there is a dangerous myth that making naloxone available fosters drug addiction. This perception is not based on science but on stigma. Second, people do not have sufficient knowledge and training about naloxone use. My research will examine the attitudes and practices of community pharmacists in Georgia regarding providing naloxone and related counseling. Are pharmacists in Georgia comfortable about providing naloxone? Do they think that making naloxone will foster addiction? Are they confident about providing naloxone and related counseling? What counseling information do they provide? Do they teach people receiving Narcan that 911 should be called and then Narcan be sprayed through the nostril? I'm administering a statewide survey with paper and electronic, and also conducting interviews with pharmacists to answer these questions. The knowledge gained from my research results will be used to design strategies such as developing training and introducing policies to help and empower pharmacists to provide naloxone and related counseling through community pharmacists. By doing so, we'll prevent those precious 65,700 lives from being lost. People we love, my cousin, your neighbor, and people with so many wonderful talents and abilities will survive to make positive contribution to each of us and our society. Thank you.
The next competitor this evening is Morgan Gonzalez. Morgan's talk is called From or With, Framing COVID Death in the News.
Ivermectin, bleach injections, essential oil, and prayer. Distrust in news media and public health officials have led to these dangerous health choices related to COVID-19, among others. What's happening here, and how are people led to make these dangerous choices instead of choices that are backed in science? To answer these questions, we have to understand a process called framing. We all realize that CNN and Fox News present the news in different ways, but the biases and the ways in which the news are presented may in fact be more subtle and complicated than you may think. Framing is a process through which the audience is led to accept a certain interpretation of facts. Journalists may not even know that they are invoking certain frames in their reporting. This means that the framing process can be subtle and almost imperceptible. Framing is an area of study in journalism scholarship that has been accepted, but I am focusing on perhaps the most crucial topic in recent decades, COVID-19. Framing is especially important to understand in terms of healthcare reporting, and this is because the news media impacts the public opinion, which in turn impacts public health decisions and public health outcomes. I am studying the most severe of these public health outcomes related to COVID-19, which is death. In Georgia alone, there have been almost 2 million confirmed cases of COVID-19 and almost 30,000 deaths. This means that this issue is critical to study, and to do so, I am analyzing a set of news articles from the beginning of 2020, the beginning of the pandemic, and from the end of 2021. I'm using a qualitative research software called MaxQDA to analyze all these almost 200 news articles. I'm looking for patterns in how death is framed in these new news articles in the hopes that I'll be able to recognize pitfalls that journalists fall into, which lead to the politicization of COVID death and public health reporting. I'm still in the coding process, but some of my preliminary findings suggest that these patterns are clear in the data set that I'm looking at. This includes the spectrum of COVID-19 outcomes, whether a news article focuses on mild illness or on death. This includes the type of person who's represented as dying from COVID-19. This can invoke different levels of empathy in the reader. The other sort of key finding I'm looking at so far includes the type of reporter that's doing the journalism. Is this a health beat reporter who specializes in healthcare, or is this a general reporter who may have been called upon to study COVID-19 and report on it? Other factors include time period, news outlet, and sources cited. I hope that my research will impact my own reporting and the reporting of others, leading us from an era of fake news to an era of trusted, informed reporting. Thanks.
The next competitor is Hallie Wright. Hallie's talk is titled Shattering Expectations, Characterizing a Novel Shattering Gene in Finger Millet.
When I was a child, I loved playing outdoors. One of my favorite things was to find a dandelion and try to blow off all of its seeds as I made a wish. Although it might be amusing seeing seeds wisp into the wind in the case of dandelions, it's actually quite detrimental when we think about crop plants. The natural dispersal of seeds from ripened plants is called shattering, and shattering hinders food production because farmers aren't able to harvest seeds that have already fallen to the ground. For this reason, shattering was one of the first characteristics farmers tried to get rid of thousands of years ago during crop domestication. Our lab sought out to find the gene responsible for shattering in finger millet, which is a staple crop grown in Eastern Africa and India. We first created a study population by breeding a shattering with a non-shattering finger millet plant, as you can see to our left. By assessing the differences in the genetic makeup between plants in our population, we were able to find a region of the finger millet genome, or collection of its DNA, associated with shattering. Then, one by one, we analyzed genes in this region for mutations or differences in the DNA sequence between wild shattering plants and cultivated non-shattering plants, and we found a candidate shattering gene. Although several genes have already been identified for shattering in other cereal crops, much to our surprise, our candidate was the first of its kind, a novel shattering gene. Because the loss of shattering is one of the key traits associated with crop domestication, understanding mutations in our shattering gene can give us a greater insight on finger millet's domestication theory. After analyzing 300 finger millet plants for mutations in our candidate, I propose a two-step domestication theory. Finger millet was first domesticated in the African highlands, as shown by 1 on our map of Africa. Here, finger millet plants partially lost, but not completely shattering, designated by mutations that separated wild shattering plants from cultivated non-shattering plants. Then, 2 additional domestication events took place in the African lowlands, designated by 2A, and in Ethiopia, designated by 2B. And in both of these regions, shattering was then completely abolished, as suggested by mutations that separated Ethiopian from non-Ethiopian plants. By identifying this shattering gene, we now have a greater idea about finger millet's domestication theory. And we also have a gene which can be exploited in other crop plants to reduce food loss associated to shattering. While you previously might not have thought too much about how that seed comes off that dandelion ahead, it's clear to see that whether a plant shatters actually matters, especially in our case of finger millet.
And our final competitor is Caroline Naron. Caroline's talk is called Coastline Redefined, Tidal Marsh Flooding, Climate Change, and Their Impact on the Georgia Coast.
Close your eyes and imagine with me for a second that you can feel the warm sun on your face, you can hear the ocean waves crash into the sand, and you can feel the warm breeze all around you. Can't you just smell the salt in the air now? The coast is an amazing place. Not only is it home to beautiful beaches like those found off the coast of Georgia, but it's also a place where 2 very different ecosystems converge. Salt marshes are an example of one of these unique ecosystems that toe the line between the land and the ocean. Part of the time, they are dry, like the mainland, while other times you can't tell the difference between them and the ocean. These differences are caused by the ebb and flow of tides. Tides follow a natural cycle that changes the amount and time that ocean water occurs on the salt marsh's surface. But not only are these places beautiful to look at, they serve a variety of functions, including providing habitat and nursery grounds for numerous species, including those that are important to our fisheries. They provide flooding and storm surge protection. They provide ecotourism opportunities like kayaking and wildlife photography. They improve our water quality, and they can even help us combat climate change. Now, this is where my area of research comes in. It's called carbon sequestration. Carbon sequestration in salt marshes occurs when plants take in CO2 gas from the atmosphere and store it in their cells. Coastal and ocean ecosystems are particularly good at storing this type of carbon long-term in their soils, and it's known as blue carbon. Blue carbon is different from carbon stored on land because the plants in the marsh can actually store the carbon for thousands of years rather than 100 years like a tree can. Unfortunately, we are losing these valuable coastal areas at an alarming rate due to sea level rise caused by human-induced climate change. For me, we are looking at how sea level rise affects certain marshes and how we can better understand their tidal systems and how often they flood. To do that, I utilize satellite data along with data from instruments in the marshes themselves to understand flooding patterns over time. Satellites, such as those from NASA's Landsat missions, go back to the 1970s, allowing us to analyze flooding patterns over long-term periods so that we can tell trends in sea level rise. This sea level rise research is very important because not only will it affect our generation, but generations to come. Thank you.
Wow, I hope you join me in recognizing what a wonderful slate of competitors we've just had. My name is Ron Walcott, and I'm the Dean of the Graduate School, and I'm so impressed with our students. Now, I'm going to turn it over to James to tell you what our students have won.
Thank you, Dean Walcott. I'd be happy to. The winner this evening will receive $1,000. The runner-up will receive $750. The People's Choice winner, which we'll get back to in just a second, will receive $500. And each winner will receive a plaque, and their names will be engraved on a separate plaque that will be showcased at the graduate school. So I want to make sure everybody remembers to click the link below so you can vote for your favorite competitor as part of the People's Choice Award. We'll announce the winners the next day on our social media and on our website. And so now I'd like to pass it over to Meredith Welsh Devine, Assistant Dean of the Graduate School, and she will tell us about our winners.
Thank you, James. It is my pleasure to announce our winners. The runner-up is Shrina Pradhan from Plant Breeding Genetics and Genomics. Shrina's presentation was entitled Salt Glands, When in Doubt, Sweat It Out. And the grand prize winner of the 2022 UGA 3-Minute Thesis Competition is Leah Dutton from Music. Leah's presentation was entitled Prevention, Early Recognition, and Intervention Techniques for Injuries in String Players. Now it's your turn. Don't forget to put in your vote below for People's Choice. So thank you all to our wonderful competitors, to their academic programs, and to you for tuning in. We'll see you next year.